Welcome to the Qualitox podcast. I'm Nicole, your host, and my guest today is Galen. Steve is a veteran in the pharmaceutical industry, both in manufacturing and in clinical trials. He has been there for 27 years. He's also an entrepreneur, and he is working on improving the way clinical trials are done. So today we're going to talk with him about what are clinical trials, is their goal, what are the steps in clinical trials, and what are the biggest challenges, and what is his solutions on how to solve them. So let's get to it. Hi, Steve. Welcome to the Qualitox podcast. Excited to have you on the show. Really honored.
Thank you, Jan. It's really great to be here. Thanks so much for the invitation.
Yeah, so I think it will be quite an interesting topic to talk about because usually I talk about the pharmaceutical industry in the GMP window during the production, during the commercial phase. So I think it would be quite interesting to go a bit back and see where it all starts. So in this episode, we'll talk about the basics of the clinical trials. So after the development where the drugs are going to do clinical trials to see if they are fit to use in humans, it's a really fascinating field. So Steve, what is your background there? How long have you been working in this field, and what are the highest positions that you held there?
Sure, Jan. So actually, I'm going to start at a place that most people might not expect, and that's that I actually started out in the industry in solid dosage and parental manufacturing. So I actually come from a very strong GMP background, and believe it or not, understanding how manufacturing works and understanding quality standards around manufacturing in pharma is very important, very useful in terms of understanding how a clinical trial works. So I gave away a little bit of my background. I started out in big pharma in pharmaceutical manufacturing. I spent about 7 or 8 years there, and then I moved over to R&D. This is all at Merck. I was at one of the big pharmas, and then spent about another 8 years in research and development. The latter part of that I actually spent in project management. And project management is the first place in a pharma company where you can actually see the entire clinical trial. It's from sort of a 100,000 foot level. It's very high, but you can see everything about a clinical trial. And then from there, I moved over to the CROs, and I was in the CRO space for about 10 years before I went out on my own.
Thank you for that introduction. So you say you are on your own. So what are you doing right now?
So I actually went out and started a software-based consultancy, and it'll be logical, hopefully, to understand why once we get through some of the discussion today. What I found is that clinical trials, as hard as this is to believe, are so poorly designed and overseen that there's a huge opportunity to improve that. And once I had been in the industry for a long time and done my detective work and did that detective work in very senior operational positions, it became clear to me that there was a need for better solutions in terms of designing and overseeing clinical trials. Again, I know to the audience that's going to sound crazy. This is an industry that I saw one industry report by 2025, the expectation is clinical trials will, the spend for clinical trials will be about 70 billion US a year. And so you'd think in something that massive, of course, everything is figured out, but it's not figured out.
Great. So maybe we will get into it and see what are the issues there and how you're trying to solve it. And as we promised to the audience, we'll start at the beginning. We'll try to cover the topic from the helicopter view and understand what clinical trials are. So let's start with the beginning. So what are clinical trials and what is their goal?
It's actually a fascinating question, Jan, because if you go to people in the industry who are designing and running clinical trials for a living, you'll get many different answers. So I'm going to distill it down to an answer that, frankly, no one can argue with. And once you have that answer in mind, it's going to help orient you as to how you have to design and run clinical trials. So the answer is the main goal of a clinical trial is to answer the question or the questions that have been posed in the clinical trial protocol. So for those who aren't familiar, in a clinical trial, the protocol is the thing that contains all the information about the study drug, as well as how it's going to be administered to the people who enroll in the trial. It'll have things in there like how many visits the patient will make during that period of time that the trial is running, but also very importantly, it lists the questions that are basically the basis for running this experiment, which is what a clinical trial is. So again, the main goal of a clinical trial is to get an answer to the question or questions that are posed in the protocol. So a lot of people, they sort of bristle at that definition. And what they say is, no, the purpose of a clinical trial is to enroll patients in the trial. But that isn't the main goal. Now, don't get me wrong here. Enrolling patients in the trial and patients in general, we have the reason we're all in this industry is because we want to help human beings have healthier, happier lives. I'm not talking about that. Of course, that's true. But if you talk about the process that is a clinical trial, enrolling patients is actually sort of in the middle of it. And you have to go back and remind yourself that the main goal of a clinical trial is to get a clean data set. That's what it's called, where all the data that have been collected are in the proper form. And you need clean data from a statistically acceptable number of patients who've completed treatment with the study drug. And then you use the data that you get from those patients who have taken the study drug to answer the questions posed in the protocol. So the problem, and we'll talk about this more later, is that the way that trials are currently conducted, everyone sort of focuses on their piece of the trial, and very, very few people are practically focused on getting to that main goal. So, of course, people understand what the primary goal is, meaning getting answers to the questions posed in the protocol. They understand that on some sort of nebulous level, but as far as having a rigorous quantitative view of how all the activities that people perform for a clinical trial impact that main goal, it's not true.
Thanks for giving the overview of the goals. And you mentioned protocols and the questions to be answered. So do all trials have the same questions, or how people come to the right questions to ask during the trials?
It's a great question. So first, the disclaimer, which is I'm not a physician, right? I'm an engineer. So you're going to get my perspective on this. So every therapeutic area and subspecialty, every study drug is different. So yes, there are always going to be significant differences protocol to protocol, but equally there's a lot of overlap or common things that are discussed in protocols. And in fact, there's been many efforts in the industry to sort of coalesce around what the basic things are, and so that the process of being able to develop a protocol is actually sped up. Now, another really interesting thing about this, and I'll try to explain it so that it makes sense, is that think of a protocol as a bunch of things that have to be done. And so the more things you put in there that have to be done, logically, the longer it takes and the more complexity there is. So actually, one of the most difficult things to deal with protocols is people refer to it as protocol bloat, right? Is that a lot of things get shoved in there. And frankly, the more things that are in there, the longer it takes, the more complicated it gets. And that also has important impacts frequently on being able to enroll patients in the trial, because as protocols get more complicated, frequently what happens is it gets harder to find patients that meet all they're called inclusion exclusion criteria, the criteria that are used to see if a person is sort of fit for enrolling in the trial. So you actually asked a really good question, Jan, and protocols are very different, but there's a lot of commonality as well. And the industry is trying to exploit that commonality again to make clinical trials much more concise. It doesn't work very well currently, but people are making real efforts to do that.
Great. Steve, can you give an example for one or two questions just so we understand what we are talking about, what the question looks like, and what has to be answered during the trials? Just a small example.
Sure. So I'm going to focus on, again, on what are called inclusion exclusion criteria, because those are very important, since if a person, prospective patient, if you will, doesn't meet these criteria, then they're not going to move forward into the enrollment process. So I'll just give two clear ones, hopefully. One that's just typical, and then one that is sort of specific. Okay. So a typical one might be body mass index, or blood pressure, something like that. Because you can think about it is that if there's a disease state that the medicine is supposed to treat, then you want to make sure that people not only have the disease, but that they also fit within a certain range, right? So, for example, if you're doing a type 2 diabetes trial, then something like BMI and or blood pressure could be very important, right? So that's sort of standard. Now, what happens, I'll go to sort of the specific extreme now, is that there are medicines for which only certain genetic subtypes are eligible to be enrolled in the trial, because the developers of the medicine have figured out that there are genetic subtypes that the medicine is more effective for. So there, what you might have is a very specific set of genetic tests that have to be performed to make sure that the person is, meets those criteria.
So those were criteria questions, basically the question that the researchers ask the patients who want to enroll into the trials to see whether they're fit, right? And there are probably different questions that are not specifically later patient specific, but on the effects of the drugs, right? So how it reacts. So more scientific questions basically also are part of those
Correct.
protocols. Okay.
That's
right. Great. Thanks for this summary and those examples. I think it clarifies a bit. And still, so still we know that clinical trials last quite a long time, and they have different phases and they different parts to those clinical trials. So can you give an overview of that?
Sure, Jan. And it's actually an excellent place to start if you want to understand how interconnected things are in a clinical trial, and how much opportunity there is to better design and oversee them. So just a note up front, I'm going to answer your question talking about the parts of a traditional clinical trial, where patients actually come into a medical facility for testing on a regular basis. Those are called patient visits, when patients come into the medical facility for testing. But everybody is probably familiar with now, especially with COVID, that there's a whole new way of conducting clinical trials, and that's frequently referred to as decentralized clinical trials. So there are parts that are similar and parts that are different when you talk about a traditional trial versus a decentralized one. I'm not going to talk about decentralized trials here. It'll be too confusing up front. Happy to address it on a later question about the parts of the clinical trial, just by sticking with a traditional clinical trial. So with that caveat, let's talk about the main parts of a clinical trial, and there are 9 of them. And each one of these pieces has sub-pieces, and that can get complicated. And so we're going to avoid talking about sub-pieces. We're just going to talk about the big parts. And the thing to keep in mind about all these things, Jan, is each part connects to the next part. And it's really, if you remember earlier on, what I said is that I was going to reference manufacturing when we're talking about clinical trials. And this is actually where solid dosage manufacturing in the industry, you know what the steps are, right? So for example, when I was compressing tablets into the final tablet form, you would get the granulation, and then you would drop it into the machine, and then you would compress it into tablets, and then you would weigh it, et cetera, et cetera. Believe it or not, a clinical trial is really not that much different. However, the industry doesn't see it that way. So the reason I'm making a big deal out of this is because if you understand the parts that I'm going to describe, and then understand that each one is connected to the next one, then you're going to see the problem that the industry currently faces. Okay, so here we go. The first thing you have to do in a clinical trial, it's something called site selection. And that's where you select the medical sites that you want to participate in the study. So these are the facilities, the medical facilities, where the patients are going to come and enroll in the study. So step one, you have to select the sites. The next step is what is called site activation. So those sites that you selected, they have to go through a regulatory approval process to make sure that the sites and the personnel at those sites are qualified to be able to take on patients in this particular trial. So if people are familiar with things like institutional review boards or ethics committees, that's where they come into play, is during this site activation process. And at the end of the site activation process, a site sort of gets a stamp of approval, and they're now ready to screen patients, which is the next step in the process. So the fourth step, or the fourth part in the process, is now enrolling patients. So those patients who made it through the screening process enroll in the study, and now those patients are ready to start what they call the patient journey, which is laid out in the protocol. It's how they'll take the study drug, how often they'll take it, and then what kind of data will be collected. So that takes us on to the next step. We're now on to the fifth step, where data are collected from the patients when they come in for study visits to that site that we've been talking about. And so the number and the schedule of those visits is laid out in the clinical protocol. Sometimes data are also collected by other means. So I don't mean to say that patients coming into a site is the exclusive way that data are collected, but it's probably the most common way. Now, the next step, it may sound sort of silly, but it's actually pretty important, is once those data are collected, they aren't automatically entered into any system, into any electronic system. So the sixth step is to take those data that were collected during the patient visits and then put those data into the appropriate electronic system. And that's usually called an electronic data capture system, an EDC system. And also try not to overcomplicate this a little, but sometimes steps 5 and 6, meaning the collecting the data and putting it into the system, are combined into one step. That's usually what happens in other industries, but in our industry, we're just making progress towards that in perhaps the past 5 to 6 years. So it's still pretty uncommon, unfortunately, but it does happen. Now we're on to the seventh step, which is to review the collected data to make sure they meet the required data standards. So this is a little different than what we've talked about previously, because what we're talking about here is data themselves have requirements. And what happens is there's a whole army of people, it's a whole profession actually, to make sure that the data that are collected meet those requirements. Because if the data don't meet those requirements, they don't make it into the part of the study where they will be analyzed. So that's step 7. We're almost there, just 2 more. The eighth part, or the eighth step, is to organize the data in a way so that the biostatisticians can answer the questions that were posed in the protocol using those data, right, that were collected. And the term in the industry is the data were cleaned. So those statisticians come in at this 8th step, they analyze the data, and they make those data available for analysis. And the last step in the process where those data are actually made available for analysis is something in the industry, it's usually referred to as database lock. So all the data have gone into a database, they've been cleaned, which is the term that's used, and now they're available for analysis, and that's called database lock, or DBL. And what's interesting is, just so that people might have a little bit of familiarity with clinical trials, steps 5 and 6 and 7 and 8, all these things, they're collectively referred to as data management. And now finally, we get to the thing that we're interested in, right? Which is to perform the required statistical analyses on the data and report the results, meaning now you're finally going to answer the questions that were posed in the protocol. So that final product is often called a clinical study report, or a CSR. So what I just want to get across here is those steps are all essentially one after the other. And if any one of them gets delayed or accelerated, then you're going to change when the data are going to be available for analysis and when you're going to be able to answer the questions in the protocol. So hopefully now, Jan, you get a sense of how big a clinical trial is, what the parts are, but also that in some sense, it's sort of like a manufacturing process wherein if I delay something somewhere, then the thing that I'm most interested in, sort of the product that comes off the end of the manufacturing line, that's going to be delayed too.
Right, Steve. So all those stages are there to answer probably the questions in the protocol. So do you take a look at the data while you go through those stages, or do you wait until the end to do it?
So they're called interim analyses. So, and there are reasons of efficacy and safety why you have those interim analyses. So yes, frequently you do stop at a certain point. Again, it's all done in a proper way so that you don't break the blinding of the study. But yes, you do stop and look at things. And in fact, there are now whole trial types that are sort of based on running small cohorts meaning a group of patients through a protocol up to a certain point, stopping, evaluating, and then making decisions based on what you saw there. So yes, that is a real part of how clinical trials are designed and run today.
Okay. And we have different phases of clinical trials, right? Phase 1, 2, and 3, right? And
Yes.
do you start the whole process and do you create new protocols for each of those phases? Or is it one continuous process?
So in general, there are different protocols that in each phase. However, the idea is that you're mining information from the previous ones for the future ones. And in fact, that's why you have the different stages of clinical development. During phase 1, you're evaluating primarily safety, unless it's oncology where it can be safety and some efficacy. But in general, phase 1 is safety. And so the information you get out of the phase 1 trials, you're going to use to, it's sort of the starting point for many of the parts that go into the phase 2 protocol. And phase 2 similarly to phase 3. So yes, they're connected that way. Unfortunately, again, what often happens in the industry, though, is that some of the transitions from phase 1 to phase 2 to phase 3, especially for similar compounds, are actually pretty similar, but they're, how to put it, a lot of times people start over completely each time and they don't leverage knowledge that's already out there, and that slows down the process of creating protocols. And I think this should make sense to the audience, since the protocol is the fundamental document that drives everything in the clinical trial. If that is delayed, then you can't even get the process started, right? So it's a very important point to make. So I'm going to give you a series of answers that you're not going to typically hear in the industry. And again, why that's going to be is because I come at clinical trials basically as an engineer and a manufacturing guy. So I see the problems from a very different perspective. It's not a unjustified perspective. It's just a very unusual perspective. And by that, what I mean is everything I'm going to lay out here as problems that are impacting trials in the way that you said, latenesses, and being extremely expensive over budget, et cetera. I'm going to explain things in a way so that you can see that what is going on is really much more fundamental than the kinds of answers that you typically get when you ask the question that you just asked. Okay, so again, a little bit of an intro there, but what you're going to hear is very different than what you usually hear. So in my mind, and engineers frequently think in lists, right? So there's 5 big problems here. So I'm going to lay them out, and then hopefully we'll get an opportunity to talk about how you solve those problems as well and what happens when you solve those problems. So the first one, and it's the biggest problem, is that really no one understands at a fundamental level how all of those 9 processes that we talked about, how they're connected from what I call a first principles perspective. So first principles means that you can go in and you truly understand what's going on, and you can describe how if I take this input and I do this to it, I'll get this output, okay? So that's a fundamental first principles understanding of what's going on. And that's the biggest problem in the industry, is people don't have a first principles understanding of the processes and how they're connected. Again, I'm just going to lay them out, and then hopefully we'll get a chance to talk about how to fix these things. The second big problem is that in the industry, people do their pieces of the clinical trial. Like I said, each one of those 9 steps has sub-steps, and there's people whose entire professions, in some cases, are dedicated to not even just one of those steps, but sometimes a sub-step in one of those steps. So unfortunately, in the industry, everybody sits on their own islands, and they don't talk to each other. So I use that sort of generous description of people sit on their islands. I have a friend in the industry who said, Steve, it's not really islands they sit on. He said, it's more like terror cells. And he was laughing because he said, it's like they're forbidden to communicate, right? Upon pain of death, what goes on in their piece to the next piece. But the problem, you can see it, is that if everybody's sitting on their own island and nobody's talking to each other, then how can you ever have a focus on what the end goal is? And that's basically the second big problem. Third big problem, even on the islands, people don't know how their process works. So and this is very, for folks in quality and manufacturing, if I talk about process capability, right? It always, people go, yeah, of course, right? If you have a process, you have to know process capability. So in manufacturing, right, you have control charts. In the old days, they were paper, right? It's been a long time since then, but you also know there are rules to control charting, right? And you can tell if your process is in control or not. Well, there's a process, right? And you understand it, and therefore you can put the control chart limits on it. Believe it or not, in clinical trials, the people sit on the islands, they don't know how their processes work. So I know, by the way, Jan, I know a lot of this is sort of hard to believe. When people hear this, they're like, how can this possibly be? But I've often described my time in this part of pharma as sort of Alice in Wonderland, because I came in as an engineer with a manufacturing background, and I honestly, every single day, could not believe what I was seeing. But importantly, like Alice, I stayed there, right? So I could figure this stuff out. Anyway, back to the problems, okay? Problem number 4, okay, in a world where you have important processes you want to understand, but you can't understand them from first principles. So what we do today is we apply artificial intelligence and machine learning to them. Now, the problem with taking an AI or an ML approach is that really those things are just correlation engines, right? They say, if this happens, or if these series of things happens, we see that this thing down here happens. Okay, that's good. If you don't really understand what's going on with your process, use that. But the problem here in our industry is that people are using AI and ML when they shouldn't. So let me try to, I'll give sort of a rough analogy to this. So if you have a black box sitting on the table in front of you in your office, and you have on one side of the black box, you have the numbers 3 and 3 and 4. No mathematical operators, just those 3 numbers. And on the right side of the box, you have the number 10. So you're going to think about it for a minute, and you go, oh, the thing in the box, it does addition. I feed in a 3 and a 3 and a 4, and I get a 10 out the other side, right? Okay. If somebody comes along, who doesn't understand how to add, they'll look at that and go, oh my goodness, there's some magic happening in the box, and there's these numbers over here, and there's this number over here, and I don't know what's going on. So let's apply machine learning to it, right? Well, that's actually what's happening in the industry. I know it sounds ridiculous, right? But because people don't understand how each one of these pieces of the clinical trial is connected to the next one and what the underlying math is, right? So they use AI and machine learning, and it really doesn't make sense. It's got a very poor predictive power, but everybody's pouring resources into this because they don't have any other alternative. Okay, so that's the 4th problem. And finally, the 5th problem. So when things go wrong in a clinical trial, and things always go wrong, they sort of go wrong in predictable ways, but they always go wrong. No one has the tools to see how the downstream processes are impacted. And obviously that's super important, right? Because if the entire reason we're running a clinical trial is to get to the clean data set so you can answer the questions posed in the protocol, then if I'm on my island up here upstream and something happens, but I can't really predict or understand what's going to happen downstream, then everybody's flying blind, and people get surprised then by latenesses and other impacts related to those latenesses. So, equally too, in sort of a more positive sense, let's say you're sitting down to design your study from the get-go, from the very beginning, and you want to do some things that you think should be able to get you done earlier. We also don't really have the tools to figure that out either. It's the same problem, just a different side of it. Because, for example, if I decide that I want to activate more sites or change the rate at which the patients are screen failing, I need to be able to show exactly what that's going to do to the time when I'll be able to answer the questions posed in the protocol. So again, that's the fifth problem. So I know that's a lot, right? And it sounds like a lot of problems. And in fact, these are a lot of fundamental problems, but hopefully we can get a chance to talk about how to solve them as well.
Yeah, so Steve, again, thanks for this overview of the issues, and it's baffling for me that as somebody who comes from the pharmaceutical production where we have those processes, everything needs to be documented, everything has a logic, and all departments has to talk to each other in order to continue further. So it's baffling that you said there are islands that don't speak to each other and don't know what's happening on different steps. So it makes it illogical and inefficient. So it would be interesting for me to hear your solutions. You mentioned that you started a company with a software that should help that. So that would be interesting to know, because you also come from the pharma and you're an engineer, so you're probably trying to apply some logic to it, and I'm really wish to hear more about it. So what is your solution, and really why is not why and nobody speaking on that as well?
So I wish I could answer the question of why no one's taking this approach, but all kidding aside, let's dive in. Very, very good questions. So I'm actually going to leave the software question for the end, because it's actually just it's a logical outcome once you hear these other things. So let's start, okay? So as you said, you come from a place where process owners understand their processes, okay? So that was how I was trained as well. So when I got into the part of the industry where clinical trials were being designed and run, especially once I made the transition from big pharma sort of project management to actually working in the clinical research organizations where the nuts and bolts of the clinical trials were being dealt with, that's where I actually took that approach that you just talked about as a process owner, and started to focus on what my processes were. So I'm going to give you a real example of this based on one of the steps that we were talking about, and hopefully this will make sense to you, okay? So keep the idea of control charts in your head, and here we go. So one of the positions I had, actually I had it twice at big global CROs, was I ran the site activation process. So that island, right, the site activation island, that was mine. And I ran that process globally. At one point I had 600 direct reports in more than 30 countries, and lots of associated employees as well. So it's a big machine, okay? So as an engineer, though, and as a process guy, and having come from manufacturing, the way I looked at it was, what are we doing here? And importantly, one of the first things you always do when you're the process owner is figure out what the cycle time is. So that's actually how I started. So what I did was I was doing a lot of things as I was first running one of these global study startup groups, as they're called, a site activation group. But what I did was we had a database. We weren't doing much with the data in there. But here's the question I asked people, here's the analysis we did, and this is what we learned. And this is going to go a long way to answering your question. So as we said, 9 parts of the clinical trial, I'm talking about part number 2. Part number 2 doesn't start until part number 1 finishes. So the start of my process, I'm trying to calculate a cycle time here. The start was when the site was selected for inclusion in the trial. So that's the end of part 1. The end of it is when that site or a site has the appropriate regulatory and legal approvals to start screening and enrolling patients. So I have 2 milestones for every site. When was that site selected for inclusion in the trial? And when was that site approved to be able to screen and enroll patients? Now it's just a cycle time. Take the later date, subtract the earlier date, get a cycle time. So you sort of go, well, like, big deal, right? I mean, right? But no one did this, right? So what was fascinating was I did that. Now you have to understand the scale of what I was talking about. I described the number of people and the number of countries. So it's called activating. We were activating 5,000 or 6,000 sites a year. And we had a database of, I don't know what it is, 20,000 completed site activations. So we just went in there and looked at the cycle times. And importantly, not just the cycle times, but we basically made histograms out of the cycle times. And what we were looking for was, or I was looking for, and the underlying math. Okay. So here's what we saw. It didn't matter what country you looked at. It didn't matter what therapeutic area you looked at. It didn't matter what client we were working with. All the data were log normally distributed, right? So they were, if you know what a log normal distribution looks like, right? It sort of looks normal on the way up and then it has a tail at the end, basically, right? And that's not surprising. Anybody who's been in manufacturing or biology or anything, log normal behavior is pretty common, but it was there, right? So that was, by the way, that was the beginning of sort of a 10 or 12 year journey, right? That got me to today. But that's what I'm talking about is on your individual island, you can figure out how your process works. And so I knew now at a very high level that my process worked and it was log normally distributed. Now, eventually I figured out why it was not log normally distributed. We're not even going to go into that. But now what I'm saying is on each island, you can do the kind of analysis that I just talked about. And then you can figure out sort of what the process looks like, and then hopefully figure out what kind of math represents it. If that makes sense, right? So now what we're saying is you can figure out what's going on each island. So now we can go back to problem number one, which I said is the most fundamental problem, which is no one understands at a fundamental level how all 9 processes are connected to each other from a first principles perspective. Well, now we have the first principle for an island. Now do that for every island, right? And then connect them and see how they're connected. Now I'm sort of waving my hand at that, but that was 10 plus years of me literally taking on every senior operational position I could in all 9 of those places, right? So that I could get my hands on the data and do the kind of analysis that I described for site activation. Now, once you do that, now what I'm saying is claiming is that I know how every process is connected to every other process. And also if I perturb one of those processes, what's going to happen to the things that happen downstream. So it's a really big claim. I get it, right? But if you follow, I mean, from a manufacturing perspective and a quality perspective, what I just described is pretty logical. So hold on a couple more minutes. I'm going to solve all the problems. Just bear with me and I'll solve them and then we'll pause. So now we solve problem number 1. So we now know how all the processes are connected, and we know that from a first principles perspective. Now let's see what happens to problem number 2 and then problem number 4. So problem number 2 was that trial execution is done on these islands and no one understands what happens on the other islands. And therefore no one understands how what they're doing is going to impact the next person. Well, we just got rid of that problem because now we can predict, right? If something happens in site selection and I'm the one who owns site activation, if site selection is late and you flow that through the kind of system that I'm talking about, I know now that, basically I'm never going to meet my original timelines unless I do something drastic to the timing of my process. And now as someone who understands what a process owner is going to say, Jan, if you go to a process owner and you say, I need you to cut your cycle time by 40%, they're going to tell you you're out of your mind, right? And yet in my industry, people regularly have those conversations because no one understands, again, the downstream impact. They make sort of ridiculous requests. I don't mean that it comes from a bad place, but people don't understand. So anyway, problem number 2, operating on these operational islands where no one understands anybody else, that gets taken care of. Now we're going to go to problem 4, the AI ML problem. Well, remember I said if you know that what's in that black box is addition, then you don't need AI and ML. And so that's what I'm saying. Problem four, and by the way, in the industry, people have a hard time with me when I say this. What I say is AI and ML for drug discovery, beautiful. Go with it. As a matter of fact, when you're looking at something as complicated as a human being, you're not going to figure things out otherwise. But if you tell me that you need AI and ML for manufacturing, you're wrong. And the problem is that tens and hundreds of millions of dollars have been invested in these approaches, right? So there's a lot of sunk costs. So anyway, we're almost done, right? So that was problem four. And now we're going to talk about problem five and fix it again with how we solved problem one, which was now we have all these processes connected and we know how they work. So problem number five was basically when things go wrong in a clinical trial, and they do all the time, no one has the tools to figure out what's going to happen downstream to that thing that I care about most, right? Well, now with problem one solved, I know how all the processes work. I know how they're all connected. So now when something goes wrong in a clinical trial, I can simply, well, I won't say it's simple. It's simple now. It wasn't simple before, but I can go into the software basically and show what the downstream impact is going to be. And also in a related way, if we're having a discussion before we ever start the trial, if we're trying to figure out a way to get done more quickly, and I'm going to say, okay, let's take this approach to reduce the screen fail rate by changing some things, for example, in the protocol. Now, because I have the answer to number one, which is this completely connected set of processes, if I change one of those processes and truncate it, then I'm going to pull back the time that's required to get to the final step, to step 9. So sorry, that was a lot of explanation, I know, but if I had to summarize it, what I'd say is once you understand process capability and you apply it to every single of those 9 processes and see how they're connected, then everything starts to make sense and you can make real progress in terms of designing and overseeing clinical trials in a much better way.
Right. So for me, from their perspective, it seems like the whole process is like in a global project manager who oversees everything and takes the data into account. So is it the issue, and is it something that basically the software helps solve by replacing a person that runs and between the islands and discusses with everything? With everyone, everything, the data, and make decisions.
Okay, so Jan, you're going to get me into big trouble here, but yeah, I'm going to answer your question. Yes, you are correct, right? So let me start out first before I say anything really provocative about our industry. I'll put on my project manager hat. So I am PMP certified, right? I'm a professional project manager, among other things. So that's where this statement is going to come from. So if you look at most industries that, and most excluding ours, right? So all industries other than ours. When it comes to project management, around manufacturing and other things, project management is no more than 10% of the total cost of the product. And that cost is borne by the manufacturer of the product, not the consumer of the product. Okay, 10%. Okay. So, and again, the person so think of it this way: you go in to buy a new car, right? If someone comes to you and says, well, you know what? Project management costs were higher these past couple months, so your automobile is going to cost more. You're going to say, what are you talking about? That's your problem, right? I'm not paying any more for this car than what I normally would. But now you can sort of see where I'm going to go. In our industry, in fact, project management costs are on the order of a third of all costs. And by the way, people on the call know something about costs in clinical trials, you're going to say, no, they're no more than 20%. No, believe me, I know where the bodies are buried. It's more like 30%. Okay. So what happens is you give things different names and it's still project management. Right. And those costs are borne by the consumer. Right. Those are not costs borne by the CRO, the clinical research organization. If you're, for example, the venture capital funded biotech, you're paying for those project management services. And to your point, Okay, so that sounds like a pretty dire circumstance. Why does it exist? It exists for exactly the reason that you said, Jan, because you have these islands, and the only way to get communication from one to the other is you hop in your boat and you go back and forth, right? And you try to make sense of it. So I'm not really, I don't mean it at all to be disparaging to people in the industry who do project management, but in fact, the reason the job is so hard is because these fundamentals that we've been talking about are not well understood.
Right. So to summarize all that, so because there are so many islands, so many issues, and it's difficult for one or a couple of project managers to deal with it, that's why we need a software that gathers the data, analyzes it, and tells you what to do, basically, right? Great, Steve. So that's great to hear that you, through the years, develop such a software, and I really hope that it will bear fruits for you. And if it bears fruit for you, I understand that it bear fruits for the industry and the companies, the CROs, for everybody, and especially the patients who get their treatment much quicker and for probably for fraction of a cost because all of delays the prices rise, right? So it's a great it's probably for me it's sounds interesting, and probably somebody who comes from the field and understand much better where you're coming from, so it may be even more close to the heart to them. And if somebody wants to catch up with you, ask you questions, and have some insights from you about what's going on in the field, and see if they can get some consultancy and so on from your side on that, how to improve their processes, what is the best way to get in touch with you?
Thank you, Jan. So best way to be in touch, LinkedIn is great. Also, I have a website. It's the name of my company is Galen FP Solutions, and now everybody knows what the FP is, right? First principles. So Galen, Galen, FP Solutions, all one word.com. Either of those ways are great, and I'd love to talk to people. Just one comment here too, because I don't want everybody to walk away from the podcast thinking that this is all about really neat software. That's only part of it, right? The other part of what you have to be able to do is to oversee the trial once it's running, and then marry up the data from the trial to what you thought was going to happen. So that's also a huge, I spent years developing that capability as well. And the thing that I want to point out, and anybody who's in the industry knows this, when you're designing and running clinical trials, you have sometimes dozens of systems that you put data in. So you have to really take that problem into consideration when you do operational oversight, and I have. But in any case, Jan, really appreciate the time, and thanks for all the really, really good questions.
Thank you very much, Steve. I wish you the best of luck with your future endeavors.
Thank you, Jan.